Dendritic cells or DCs are key immune cells linking the innate and adaptive immune system, while DCs are heterogeneous. This method helps us understand which genes, including transcription factors, may regulate DC development. This technique provides a simple and quick way to identify a gene that controls DC development from their progenitor in vitro.
To begin, maintain the immortalized hematopoietic stem and progenitor cells, or IHSPCs, in complete rPMI 1640 medium, supplemented with 100 nanograms per milliliter of the FLT3 ligand and 1 micromolar beta-estradiol. For lentiviral transduction, plate the IHSPCs in 12 well plates at a density of 1 × 10 to the 5th cells per well and 1 milliliter of complete medium containing the FLT3 ligand, beta-estradiol, and polybrine. Then, add the lentivirus carrying the short hairpin RNA in each well at a multiplicity of infection of 100. Next, spin the plate at 1100 × G and 37 degrees Celsius for 90 minutes. Then incubate the infected cells overnight at 37 degrees Celsius. The following day, remove the polybrine by collecting the cells into 15 milliliter tubes and centrifugation, following replacing the media with fresh complete medium containing the FLT3 ligand and beta-estradiol. After an additional 24 hours, add 6 micrograms per milliliter of puromycin to the medium to select the infected cells. Replace this